Thank you all. We had this beautiful introduction to motors and the role of motors in neurons from the first talk from Andrew's very lovely talk. So generally carrying on the same theme, transport in neurons is extremely important because cargo needs to move from the cell body and the business end of the neuron where a lot of the action takes place at synapses. The reason you need a lot of robust cargo transport using mechanisms such as motors is because majority of the cargo biogenesis actually takes place in the cell body. Therefore, you have to cover these long distances and move cargo in the anterior direction from the cell body to the synapse, typically using kinesin motors because the microtubules are oriented such that the plus end face outside. And in the retrograde direction, clearing the synapse of things that it does not need, because this is a space-restricted system, even though I've not drawn it as such, using the dynein motor to carry things back to the cell body. What is very clear is that long-distance axonal transport seems to be very important, and when it's disrupted, you find, basically several types of human diseases affecting neurons. One of the examples that Andrew talked about was the lower mice, which is neurodegeneration taking place. But the early steps in blocking down retrograde transport is actually that the transport goes down slowly with a dying back neuropathy. So if you look at a major cargo in neurons is synaptic vesicles. They are formed in the cell body. And if you look at the path of movement, you see that, first they have to be formed. They recruit motors, which are these little pink circles. They need to enter the axon, and then they move processively on microtubule tracks. Microtubules are present usually as nested with neuron-specific lengths, typically. So obviously, when they come to the end of a microtubule, they fall off. They have to find another microtubule where they then come to the synapse. So the question that I set out to ask now several years ago in conjunction with a theoretical physicist, Gautam Menon, who's in IMSC and who's not here today, was sort of inspired. I would say it's much more of a neuronal naturalist approach where we just go and explore what we see and try to explain what we see, right? So the question was a very simple question. If you're moving along neurons, how do you maintain cargo flux in the axon? Is that only motors? Do you need other things which control it? Because some years ago, Frank Perez did this very beautiful experiment where he removed all the cytoskeletal elements and then watched in non-neuronal cells cargo forming at the Golgi and tracked how long it took to come to the cell surface. In about 20 minutes, a vesicle that came from the Golgi, which is drawn over here, was able to make it out to the cell surface without microtubule tracks or actin. That told you perhaps diffusion alone was sufficient to carry cargo at least on the distances of 10 to 20 microns. Now, would that work in a neuron? And the answer is a resounding no. Some neurons, like your neurons or my neurons, who start at the back of our spinal cord and go up to our toe, can be up to 1 meter long. But the critter that we look at, C. elegans, has very short neurons. And even we ourselves have some very short neurons, which are about 500 microns. If you remove the motor, which can transport these cargo, you can wait forever and absolutely nothing reaches the destination, which is the synapse, which is somewhere from 20 to 400 microns away. That tells you clearly that diffusion alone, in neurons at least, may be insufficient, even given sufficient time, for cargo to get from the cell body all the way to the synapse, okay, to cover any distance. So why might this be the case? What's so special about neurons? So here I'm going to show you a series of electron micrographs which come from different labs. There are two possibilities, right? One is the cargo itself is very large, the neuron itself is very narrow, the axon is very narrow, or it's filled with things. And I think it's going to be a combination of all of these three. So if you look at Hirakawa's beautiful EMs, you see that cargo come in different sizes, but you can see that it's very cytoskeletally rich. So potentially, these are all going to be very crowded environments through which cargo have to make their way. And remember, cargo do need to get to the synapse at regular intervals. You can't make, for instance, a synaptic vesicle just at the synapse. You need supply from the cell body. When you look at the neuron in a small critter like C. elegans, which we study, this is the neuron that we typically look at. Look at the number of microtubules. Just the microtubules themselves fill up all the space. And you look much more closely, you see this meshwork-like structure that is present between the plasma membrane and the microtubules, making it a very challenging environment to walk through. I should be using this. All right. Now when you look at a series of electron micrographs through the neuron, you see here's up there. Like this is one series, and you might say, oh, we could fit a cargo here. And you look here, and there's much less space. And most, this is the diameter, this is about 200 nanometers. Most cargo anywhere between 50 to a micron in diameter. And you sort of wonder, how are they going to fit in this restricted space? Finally, it has been shown that you have actin-rich regions in vertebrate neurons by some very beautiful electron microscopy. And I want to say that the movement of cargo in neurons is in an inhomogeneous environment. Now here are Drosophila neurons which don't nearly look as crowded at this particular C. elegans neuron, or this neuron doesn't as look crowded as this particular neuron, which is a different neuron, and definitely there's much more space. So the picture that emerges is that you have short spurts where you can have open spaces where perhaps movement is free, and other places where things perhaps cannot move. So if you look in C. elegans in this particular neuron, which I have done EM myself, and there are lots of electron micrographs available, or if you look at vertebrates, you don't find things in between. You only find them on the surface. So I think that this bundling that occurs is partly structural, especially in invertebrates. You don't have neurofilaments to give you structural stability, so it has to be microtubules, which is anyway, as you know, a stiff polymer. In neurons, typically microtubules are not very dynamic. In fact, rock solid would not be the wrong thing to say. Very little dynamicity. Anything else? So then how would cargo move in a neuron? The best thing is to actually go ahead and look at it, which is precisely what we did. Here's a little worm. We trapped it in our little imaging chamber. We turn on light, and then you will see cargo moving in both directions. You will see a lot of cargo which don't move at all. Here's a big clump of cargo which doesn't move. Here's another big clump of cargo which doesn't move. Here's a small one which doesn't move. So as much cargo moves, there's a significant portion of cargo which is stationary. This is not only our observation. Any person who's ever looked at any paper which looks at cargo transport and images cargo transport in any system, in any neuron, you see a large fraction of cargo which are stationary, right? So when you see this large number of cargo that are stationary, and I'll just briefly describe how we do it. I mean, Andrew already set up how you look at a kymograph. You just make a kymograph where you have the distance indicated on the x-axis and the time indicated on the y-axis. A moving vesicle is sloped, and a stationary vesicle is basically looks like a rigid line. When we started looking at this data, we noticed that actually cargo was stationary for different lengths of time. We made long movies for up to 5 to 10 minutes, and we saw sometimes cargo was stalled at a particular location for as long as 5 minutes. That's a very long time. Typical pause durations are like 2 seconds, so you can see how much longer the cargo was stopping. So we decided that we would apply a cutoff. We would say that anything which pauses for more than 15 times what is the longest pause duration that we see, we'll consider them a stationary cargo, and they're present over there for a really long time. All right. Then we went about characterizing the system. We did both C. elegans and Drosophila, and occasionally we sort of foray into cultured neurons, but here I'll just show you C. elegans and Drosophila. We can count how many vesicles are stationary, which is precisely what we are showing you here. These are 2 different neurons of a similar kind, two different types of cargos. This is a different neuron in C. elegans, this is a different cargo, and this is Drosophila. In each case, we find a large number of cargo are stationary, and they're stationary for very long periods of time. So now that we have an assay and we can put some numbers to things, these are all wild type. In fact, I'm not going to show you very many mutants at all in this. In fact, none, I think. Okay. So we asked a very simple question. If the idea is that there might be regions in the neuron, for whatever reason, where cargo stall, you might expect, if it is nothing to do with the properties of the motor that move it, you might expect multiple cargo to pile up together. Is that really true? And the answer is a resounding yes. So here I'm showing you data where we look at the very abundant synaptic vesicle cargo. You see several puncta. This is several puncta. Rab5, which is an endosomal cargo which doesn't come out very much. You can find that cargo present together. This is an example from Drosophila. We can make chymographs here with mitochondria and synaptic vesicles. And what we see is that cargo often pile up together. And if you look at a low abundant cargo like mitochondria, in fact, where mitochondria are present, every instance, practically like 99% of them have synaptic vesicles stalled next to them. It suggests that multiple cargo are actually present together. The other thing we started noticing when we looked at our movies, when Parul was looking at our movies, was that, these cargo would be stationary for long periods of time. Then they would move away. New cargo would come, but they would be stalled at the same location. And this happened in 40% of the cases where the vesicles moved away, would again after, maybe, about 20 seconds have other cargo come and jam there. That gave us the idea that potentially there might be locations in the neuron which are more sensitive to cargo stalling. So obviously, the one component which is common to all neurons is both microtubules and actin. Today, I'll describe the data that we get from actin and not microtubules, and we can discuss it later if you so choose. So we just did very simple. We had two different markers. Here we're using choronin, but we've also used eutrophin. And we just image them together and say, do you see actin enrichments where cargo stall? And we can do that very simply by using these same kinds of chymographs, drawing lines where they're present, and counting all of them, just counting overlap. Simple experiment, right? And what we see, whether we look at Rab3, which is synaptic vesicles, an endosomal marker, mitochondria, or Rab4 in Drosophila, and here's a blue arrow, and this is the part I want you to see, that majority of cargo stall where you have actin enrichment. So UTCH is a much better marker than COR1, which is, it doesn't mark actin as abundantly in the neuron. So we basically concluded from this that stationary cargo are susceptible to forming or are susceptible to stalling at actin-rich regions. Right? It seems like a reasonable inference from these experiments. So what do we have? We then have a prediction. A prediction is a very simple prediction that if you can alter the cytoskeletal density acutely, you should be able to change the density of the stationary cargo. And all we did for this experiment was to inject the animal with latronculin A using different concentration. And we see about a 30% reduction in choronin or a 30% reduction in eutrophin. Everything doesn't go away. And we see a concomitant similar reduction in the number of cargo that are stationary. We see this both in C. elegans as well as Drosophila, suggesting that when you dissolve this actin, these vesicles that are stalled maybe can begin to move again. We also see this in another way, and I'll come back to that later. So the other interesting thing that we see, which is consistent with our observations, is that the fraction of vesicles that move when we remove the actin or reduce the actin increases compared to the fraction of vesicles that are stored or stationary. So then we moved on to the next question, which is if you are in this region, which is very crowded, and some of you have traveled in Indian roads, you know that there's a lot of crowding. There are people who are walking on the side of the road, you have little cycles, you have big objects, Perhaps not when you came here, but if you go to a normal Indian city, you see a lot of complex things happening over there simply because people are occupying space on the road. So when you have a vesicle coming into these kind of crowded regions, how do they behave? So you ask a very simple question, right? What do cargo do when they come to a region where you see other stalled cargo or to actin-rich regions? Do they act as traffic jams? Right? Because we've not made the case that they act like traffic jams. Here is the first time we can ask that question, whether these regions are regions that are susceptible to forming traffic jams. So the first thing that we saw to address this question is that when we look at where cargo stall along the neuron, most of the cargo that stop for any length of time, we're not talking about pausing here, occurs at these places where they already have stalled cargo. Very little stops away from these locations. That's very suggestive. The second thing that we did, and Parul did this very nice analysis not that long ago, where we sort of break down the neuron into areas which don't have any actin, areas which have an actin enrichment, areas that have a vesicle but no actin enrichment, areas that have actin enrichment as well as stalled vesicle. And what we see, and we do that both in C. elegans and Drosophila. I'll walk you through the C. elegans data. The Drosophila data fits the same thing. Where there is no actin present, most of the vesicles that come into that region just move, right? So there's absolutely no obstruction to their traffic. They can just go past. Very simple. When you have actin, these are these small filamentous structures which are enriched. It could be deep within the neuron. It could be between the cortex and the microtubules. Regardless, still majority of the vesicles do go through, but there is a little bit, 84% of them go through, but the gray bar, which is the stop bar and which is pointing to the red, some vesicles do seem to halt there. When you look at regions where there's just cargo stalled, but there's no actin enrichment, these are fewer regions. They're about 20-25% of the region that you see without an actin enrichment, a big vesicle stalled. You find that, again, a good something like 60% of the vesicles stall over there. And finally, when you have actin enrichment and a pre-stalled vesicle which is present in that region, that is just absolutely dramatic. Majority of the vesicles are unable to go past that region. In fact, they stall over there. So the inference that we do from here is the following. One, actin enriched with stationary cargo or stalled cargo acts as the strongest crowder and prevents cargo movement through that region. Two, yes. So that's, we usually say cargo crowding creates traffic jam. So when we say a traffic jam, what we mean is that cargo which is entering that region do not move fast, or most of them do not move fast, or most of them stall. That is what we call as a traffic jam. So I guess not. That is a very subtle distinction. No, a jam and because all traffic jams are dynamic, in that vesicles can emerge from it and as well as get trapped in it. The size of how many vesicles are jammed in that location seem to vary when we have neurodegenerative models, and the number of vesicle stalls is never more than 4. So it really, we could call that a congestion and we could call that a traffic jam. Either is fine. All right, so the first thing is that actin-enriched region with a vesicle stall there has the strongest probability of stopping any vesicle movement. But just a cargo stall there alone is sufficient to reduce movement. So the way we think about it is that physically the cargo must be occupying a large amount of space and preventing vesicles from going through. From looking at our earlier data where multiple cargos stop at the same location independent of each other, we think that suggests that this is purely based on physical crowding and does not have very many molecular mechanisms associated with it. So I'll just show you a movie to sort of illustrate this point. Here the green is going to be the actin-rich region and the red is a synaptic vesicle cargo. You will just see a vesicle that moves and then stops. It's pretty obvious actually. That's an actin-rich region. That's a vesicle that moves. You just stop over here. What we typically find is vesicles that stop in such regions stop for typically twice to thrice the time that an average vesicle stops, and it stops outside of such regions. So there seems to be that it just sort of prevents it from moving ahead. So if our hypothesis is right, a couple of things should follow, right? Any way that cargo is stalled should locally control flux. This would be our expectations, and that is what Parul went on to test, where she looked at regions which had stalled vesicles to see where the vesicles moved away. The number of vesicles approaching such areas across time do not vary, and what we find, it doesn't matter which marker we look at, or whether we look at anterogradely moving vesicles or retrogradely moving vesicles, as long as the stationary vesicle has moved away, you find that the number of vesicles that move through that region significantly increase, consistent with the hypothesis that removing physical crowding locally can locally affect flux. The other thing which was a prediction that we had is that if you have a lot of stationary vesicles present, a moving vesicle is not going to be able to get very far simply because of these physical crowding constraints. Is that what we see? And the answer is absolutely true. Depending on the density of stalled stationary vesicles, the run length correlates with no vesicle and on average getting further than 5 microns. And this is very significant because majority of the neurons are much longer than 5 microns. The neuron that we look at is 400 microns, and you're not getting very far with an individual vesicle, and therefore how you control cargo flow in the neuron is going to depend on how many physical constraints there are to its motion. So this is a simple model where we see that actin-enriched regions have the slowest ability to stop. Where there is cargo crowding alone, there's slightly increased ability to stop. Where you have actin as well as cargo crowding, the most stopping occurs. No, so we looked at one neuron, which is a commissural neuron, which curves along the animal. So at each point it's bent because the animal is round. We don't have that morphology, okay? But in the curved neuron, we don't see any difference in stalling. So we don't know if there are additional physical kinks will they count. In fact, we're looking at a particular place where the neuron sends out a branch, but that regulation seems to occur very differently from here. Physical crowding does count, but that's not the only thing, yes? That is something we cannot, if we don't see it, we have to assume it's not there. So yes, you can say that it's so low and we are not picking it up. That is a possibility. Now let me tell you one more experiment, which is, so we initially thought that might be trapping based on myosin anchored to actin. So we looked at myosin 5, which is known to be present on synaptic vesicles. Absolutely no change in the behavior of vesicles in these regions. So you look at myosin 5 mutants, your prediction should be if they are involved in trapping over here, then in a myosin 5 mutant, more of them should go past such regions. They don't. That's our only other hypothesis. So that's why we favor physical crowding. So we haven't done 1 and 2 because we don't get enough events because these are not, but we can tell you this is what we've looked at. We've looked at areas where the actin depolymerizes completely, right? You have cargo present over there, which are still stalled. Only in half of the cases do the vesicles all clear. And in all of these cases, the half, less than half, it's about 44%. And even in the ones which clear, they have like a couple of vesicles, not more than that. If you're more than 2 in that location, they just don't clear, and actin reforms over there. So we don't understand that's why we favor physical crowding over other things. Yes, it's still correlative, but it doesn't matter. The remaining actin, so when we treat with latrenculin, there are other factors which can lead to stalling. When we treat with latrenculin, we reduce the amount of actin hotspots, and now stalled cargo which occur outside of actin hotspots are increased vastly. We think they might be microtubule ends, but we don't know. We don't know for sure. All right, so that's where we stand with physical crowding that we think leads to crowding. So in vivo, so I can tell you we've not done this experiment. So this is going to be an experiment which my physicist colleague can say, you're using a mutant, you don't know what it does. Okay, so I'll still give you the answer. We have a mutant which makes large tubes. Okay, and what we see with large tubes is they are much more flexible. You can see the front end of the tube sort of behaving slightly differently from the back end of the tube. These large tubes, perhaps because they are more flexible, are able to go through. So now let's look at mitochondria. Large objects, as far as some of them, of the bigger ones, are more bendy, and the smaller, more compact ones are less bendy, and in fact, Steve has also published on that. The longer, bendier ones can go through where other mitochondria are stalled, but the shorter, more compact ones can tend to get stalled. But we are looking at light microscopy. There are obviously caveats to these kinds of measurements, and the general principle still holds true. Now, let's step back. When you look at large objects like mitochondria, which are stalled, and endosome-like structures which come over there, every mitochondria traps vesicles next to it. So we think that all of this data is internally inconsistent. Now, it's very difficult to perturb physical crowding inside the neuron short of reducing the density of actin. We've also reduced the density of microtubules, and we see a reduction in number of stalled vesicles. So all of that picture that comes up is that physical crowding is certainly very important when it comes to regulating cargo flow in the neuron. Okay. Are there ways to overcome traffic jams? And this is a part of the talk where we do not have an answer at all. But I'll tell you some of the things that we were thinking about. So me and Gautam kept talking about it and said, he said, let's just build a model with sort of benchmarked on some of our data, very limited data. We said these are the types of vesicles we see, this fraction of vesicles move in the anterograde and retrograde direction, and we run a simulation. What do we see? Right? And so when Gautam and his student did this kind of experiment, this is what we saw, and this is them playing a movie of his simulation. And it very intuitively makes a lot of sense, right? You have vesicles that come, if they halt, they can't do anything over there, right? Because they're walking on a single microtubule filament. Eventually, all cargo transport is going to halt unless something can happen, right? So what he puts in is that it says that when you come to halt, you can actually do one of two things. You can either change your microtubule track, which we cannot assess inside in vivo, or you can do a reversal. And then you calculate from these kinds of simulations, and you can look at the following, right? So this is the number of vesicles that can move through a region where you are able to do state change, which is switch to another microtubule or do a reversal. If you take off the ability to either switch to another microtubule or to reverse, the number of cargo that moves through a region in axon really stops. Yes? So retrogradely moving vesicles tend to stall a little bit less at stationary cargo compared to anterogradely moving vesicles. When we look at a dynein mutant or a dynactin temperature-sensitive mutant, we see that this now is exactly like the anterograde motion. So the way we explain it is from all of the data that exists in the field, including Steve's and Roop's, that dynein is a more versatile motor, and therefore this is consistent with that. So I didn't show that data. Okay, so at least it appears that reversals are going to be very important to do this kind of, to be able to allow the vesicle to sample that space again or move past what might be a congestion or traffic jam. Right? So what he sees that if it goes back, then there is a probability that it can go, in the simulation, these are the data that he sees. The way we think about it experimentally, that if it goes back, then there is a chance that it can engage with another microtubule. It has to engage with another microtubule in the simulation if it has to get past that traffic jam. In this case, he has labeled it as individual microtubules. In vivo, it could be another protofilament, it could be another microtubule. We do not know.
The microtubules themselves
So what we find is that in this cell, when we reduce microtubule density, the density of stalled vesicles goes down. So we think that it's space filling, which is very important. You have so many microtubules. Because there are neurons with far fewer microtubules. In fact, I showed you that other picture, which was chock full of microtubules, next to it a neuron which has one microtubule. So you would have, I think in those cases, we can't measure it. Possibly, it would just hop back on the microtubule at some other time, maybe locally diffuse for like, a little bit, and then try and sample another microtubule. So in mutants which have lower density of microtubules or on colchicine treatment.
Interesting question that would be, in those cases where the stalls take longer to resolve with no lower density.
We have not done that experiment. Okay, we have not done that experiment, but behavior of vesicles that come up to that point is identical. That is, as soon as a vesicle comes to a place where there are cargoes stalled, they stall with very high probability. So we decided and attempted to look at reversals in vivo, and we do that very simply by bleaching stationary cargo and watching where cargo reverse. And we see that vesicles do reverse when they hit stationary cargo. But then Parul did this very nice experiment where I should tell you upfront that only 15% of the vesicles in either direction reverse at all. So this is very different from the kind of saltatory motion that many cargo exhibit. Very, very few synaptic vesicles reverse in either direction, whether they're going to the synapse or coming back from the synapse. But what Parul saw, that the number of vesicles that reverse, which are at stationary cargo or elsewhere, are essentially the same. So this idea that we had that, when you come up to stationary cargo, some mechanism is triggered, which will now allow you to get past this congestion, and reversals might be one, is still up in the air. We do not know what is happening as of now. Okay, and I'll end this small section of the movie by showing you a video of cargo reversing throughout the axon. So again, green is actin-rich regions, and the red is the cargo. See that they reverse. You'll see that cargo also reverse in other places. In fact, they do funny things like reverse right here, go back and forth, go ahead, many interesting things. But this kind of movie can actually fool you into thinking that reversals occur at actin-rich regions. And in fact, we spend a lot of time looking at this to see whether reversals occur at actin-rich regions and what would be the molecules that would trigger it. But essentially, we do not see a correlation. Reversals, very few vesicles reverse, and they reverse in random places in the neuron. So what that means in terms of regulating cargo flow, we do not know. We do know that for vesicles to emerge from stationary cargo or to effectively stop at stationary cargo, you certainly need motor activity. So if we look at a motor, anterogradally moving motor, which is shown in a mutant of that motor where you have far fewer motors on the cargo surface that an earlier study of ours had shown, in such a mutant background, a vesicle that comes to a region which has stalled vesicles tends to stop more and fewer emerge from it. So we know motors are involved, but the predominant effect seems to be that of physical grounding. So come to the last, which is more interesting, I'm a biologist, part of the talk, is what this actually does for you in a neuron. It gives you small pools of vesicles throughout the neuron. In fact, I would go so far as to say that the number of vesicles that seem to be from our estimates of counting and from looking at synapses and knowing how many vesicles are present at this particular neuron synapses, the number of vesicles, synaptic vesicles along the axon are about 3 times what is present at the synapse, at a conservative estimate. That means you have these pools of vesicles that are present along the axon, which perhaps can act like reservoirs and can have functional consequences. So Parul, my students, spend a lot of time trying to work this out, and I wouldn't say that we've answered the question, but I think we've gone some distance. For this particular set of experiments, we look at the touch receptor neuron, which is what most of the movies that I've shown you here are today. This senses gentle touch, so we can touch a neuron repeatedly. We know that the animal habituates at a certain rate when it touches. It's a very simple experiment. You touch the animal, it moves in the back, moves forward. Touch the animal in the towards the head, it moves backward. We either use that assay, or we use an assay where the animal stimulates by putting them in little dishes which have PDMS pillars, and it just thrashes and randomly touches these pillars. In both cases, you're giving it touch stimulation, and what we saw was the density of stationary vesicles or stationary synaptic vesicles drops in, after 10 touches, after 60 touches, or if you use a PDMS pillar device, we see the same reduction. And the number of vesicles that were stalled in each of these locations goes down. So obviously this was very interesting to us. We wanted to know what might control this, and whether we will be able to use this in the future to understand how vesicles are recruited from such locations. I will show you some of our earliest data in this direction. Very, very simple data. We looked at the following mutants. We looked at channel mutants, which are abrogated in the ability to sense touch. So these are touch insensitive channels. We looked at 2 different touch insensitive channels. And when you don't have the ability to sense touch, you do not, when you do this touch assay, reduce the density of stationary vesicles. Essentially, vesicles don't move. We find that for neurotransmission to occur, you first sense touch, and then you open calcium channels along the axon to let in calcium. And if you block these calcium channels, because without which you won't have transient potentials, you find that the ability to drive vesicles away from these stationary pools goes down. We look at animals which are not able to transmit information, and this is very intriguing to us, and we're still following that up. We see the same thing. And finally, when we have a loss of function motor, which is data not shown here, we essentially looks like any of these others. But when we have a gain of function motor, which we think can move faster or can recruit vesicles, can drive transport much more, we find that you lose a lot more vesicles from these locations when we stimulate. So essentially, the stationary P vesicles recruiting from those locations depends on the ability to sense touch, ability of the neuron to allow calcium influx, the presence of the motor, and ability of the vesicles to release at the synapse. So I come to the end of my talk. I haven't taken my full hour. So it comes up, I think, what we learned from the study, what Parul and I and Gautam learned from these studies, that there are micro niches along the axon. There are niches where you have actin-rich regions, and you have cargo. You can have cargo stalled at the ends of microtubules, which I haven't described. Cargo stalled in other places, which are not actin-rich. Cargo stalled at ends of microtubules, which also contain actin, as well as just actin-rich regions. So we think that there are common constraints to all cargo motion, independent of the type of cargo. It doesn't matter whether you're an endosome, a synaptic vesicle, or a mitochondria. Long cargo halting arises most frequently at actin-rich regions, but we think cargo crowding is a significant factor in those locations. And locations where cargo becomes stationary act as local sites of motion regulation. So what we see is that when vesicles have to sample this environment, one of 3 possibilities, they're walking on a different microtubule at all, and they go past. We think in vivo, working on a different protofilament may not have a very strong effect. Alternately, they could switch tracks. We don't understand very much about it, and in vivo it's very difficult to assess. And the most common frequency, as much as 80% to 90%, depending on the location, really halt. We think that reversals, and we still don't know, might be a small fraction of vesicles might be doing reversals to try to sort of get past these stationary cargo locations. We are looking at a neurodegenerative disease model in C. elegans to see if that is the case. So the ideas that are there, which we still need to, the implications of our study, is that the ability to clear roadblock and maintain steady flow may depend on stall motion or an ability to change microtubule tracks. We think that the physical block of transport may be relevant for neurodegenerative disease, because if bigger and bigger blocks build up, as has been shown in other models of neurodegenerative disease, perhaps you just physically occlude the ability of cargo to get past such locations. And our most exciting finding is that small reservoirs of vesicles along the axon seem to be recruited dependent on the ability to, dependent on neuronal activity and motors, and this we think might be functionally important. So I come to the end of my talk. This work was started by a former postdoc and now finished and really fleshed out in all of its glory by Parul, who's a graduating student. None of it would have been possible without a lot of conversation between Gautam, who's my collaborator in IMSC, and these are the people we got stuff done. Thank you.
I mean, you try to convince us that this stalling is due to physical crowding.
Yes.
So basically, the physical conditions that you have inside the axon. And then you showed data that when you stimulate the axon, then you can somehow activate this stalled cargo. And I cannot really imagine that the cell changes the physical conditions of the whole cell.
In fact, actin doesn't change. So this happens, so we have a theory. It's a theory which we are testing now. What we think is that when you touch stimulate, you have the voltage-gated calcium channels open. Calcium now comes in and in some way activates the motor. So the motor, which is already can do much more, if the motor is not present, none of this happens. So we think that the motor must be reaching an alternate state of some kind through phosphorylation, or maybe more motors are recruited at that region, we don't know, and it can put itself out. So many more cargoes seem to emerge from those locations.
So the idea is you overcome the physical conditions by applying more force.
Yes. And we do see that because when we look, in the normal case, when we look at a mutant which has fewer motors on the cargo surface, many more of them stall and fewer emerge. When we overexpress the motor, nothing changes. But when we overexpress the motor and do the stimulation, now many more emerge from the stationary cargo. So we know that the emergence is somehow controlled by the state of the motor.
Okay, but that could also mean that actually what you need to get this jammed position is just something that starts the traffic jam. And that could be some cargo that for whatever reason stops moving, and then the thing amplifies because more and more cargo piles up.
That is precisely what we are saying.
That's what you're saying.
That's precisely what we are saying. So I haven't done a very good job clearly in my talk, in that cargo stalling itself seems to be sufficient to stall other cargoes, but the most number of cargo stalling occurs where there is actin. So the way you think about it is, this is your straight road and you have little obstacles, then obviously you will have slightly greater probability of a vesicle stalling here than when you don't have these obstacles. And once you're stalled here, that cargo now nucleates other cargo from stall.
So along those lines, I mean, a nice sort of model of this, since you've shown that mitochondria obviously can clog things up, and you know that the mitochondria, Miro and Milton, such are controlled by calcium, right? So there's a nice potential model is that, right? That you release your mitochondrial blocks.
Exactly your idea. We look, there is one clear Miro, there's no Milton in C. elegans. There's one clear Miro ortholog, and there are two other Miro-like genes. So there's 3 Miro genes. We've looked at each of those independently, and we've looked at double mutants, and there are plenty of mitochondria in the axon. They don't seem to do anything. They're different from wild type. So we've actually written a paper which we have sent out for review where we say that Miros may not be so important for getting, so, C. elegans seems to do it, yeah. So we do not know that. But there are other things which can happen too, which recognizes at least synaptic vesicles, does so by binding to PIP2. An entry of calcium can itself cluster PIP2 on membranes, and that in turn might be able to recruit more motor. These are all hypotheses, some of which we can test in vivo, many we can't.
I may have missed this, but why do you think it's important that the vesicle distribution changes with repeated stimuli?
So,
is that giving an advantage to the neuron?
So I don't think I put in all the data in this talk, but I'll just show you how we think about it right now. What you see in, I seem to have done something with. Okay, that's the curve of habituation that you see, right? You keep stimulating the animal, and this habituates. And then you don't habituate, you still show touch responsiveness after 60 touches or 70 touches. You still have 50% of the animals responding. Why is that the case? This is not the case when you look at chemosensation as much. You will drop down a lot more. So one of the ideas that we had was that the ability to keep some amount of touch sensation going, and this is a soil nematode, and touch is going to be an extremely important sensory modality, is possibly by getting vesicles to come up to the synapse. And that's the hypothesis we are testing. Consistent with that, we see that when we overexpress the motor, this entire touch habituation curve sort of shifts up. Okay. So that's just consistent. We don't know the full picture.
And so just to follow that, have you seen the direction of movement after the touching?
We are doing that. That has been difficult because you realize that these are going to be very, very small changes in flux because we only have 2 to 4 vesicles. We're still trying to develop methodologies where we can measure that accurately. And we're not sure. So we've tried to few simple things which don't show big changes, but we're not convinced that there's no change yet.
Sandhya, I have a question. In cells having protrusions, for example, transition zone in flagella or axon initial segment in neurons, there are cytoskeleton networks. So how are those networks different from these actin patches where they allow megadalton trains and all kind of vesicles filtering them into
in terms of transport, we think of it as just density. So what we also see is that the earliest part of the neuron, which is presumably the axon initial segment, we see many more cargos stalled. And despite having some amount of limited microtubule dynamics, the actin seems to be a bit thicker there, but not a great deal. So certainly the earliest part of the axon is something which we actually avoid to look at these kinds of issues because we think there we can't distinguish clearly just physical mechanisms, but yes, they do stall a little bit more there.
As if you're viewing these stalled vesicles as basically storage reservoirs for synaptic vesicles that you're going to need, my suspicion, although I haven't done any modeling of it, is that it's probably not optimal to have them uniformly distributed along the axon. It probably you want them to sort of be more common, close. So it's going to depend on sort of, variety of things.
That's an idea that certainly occurred to us. And what we find is that closest to the synapse, we find bigger clumps. We don't know what that means, right? Because we really honestly don't know. But it doesn't matter where the clump is, it still behaves the same. Right. It stalls cargo. But we do see bigger clumps. That's also the place where, because it's curved, the neuron has a little bit of a kink, so we don't know if it's because of the kink. We just really don't know.
But in principle, one could do a model where you could calculate sort of what would be optimal. So you probably, you'd have to say how many typical touches are you going to have to respond to, and you'd have to make some assumptions, but then
we'll have to make some assumptions. So we are trying to, with Gautam, do a 3-compartment model where we look at things closer to the synapse to begin to understand that, including how does flow towards the synapse happen, because this is a purely, which is unusual for my lab, a more biophysical story. We usually do much more for biological story.
Yeah, hi. So my question is related to Steve's, so regarding the Miro mutation. Yes. So have you ever tried mutating syntrophin?
There is no syntrophin in C. elegans.
There is no syntrophin. Okay.
But I have to say, when the mitochondria are moving, we don't see cargo stalling next to a moving mitochondria because the mitochondria is moving. Yes, we don't see that. We don't see that kind of, because we don't have the kind of resolution. That was done by EM, right? It was very beautiful. We don't have that kind of resolution. All we can tell you is that we see a certain density of actin, but it's sort of variable, slightly variable, and some animals there's more, some animals there's less. Absolutely, agree, it can be. But there's also been shown by Shubhajit Roy's group that there's deep actin. So we can't distinguish between the two with our assay. Anyway, the neurons are much smaller than vertebrate neurons, much thinner in diameter. It's about 300 nanometers at most.
Sandhya, this may be a very naive question. So the cargo that is you're observing, so in principle it could carry also some myosin, and then the actin, instead of being an obstacle, could actually, be there for transfer of cargo from the actin, from the microtubule to the actin. So is it absolutely certain that there is no myosin?
That's a beautiful hypothesis, right? And that's the one we were very attracted to. So we actually looked at things, that's why we looked specifically at the myosin 5 mutant. It's a null. Myosin 5 is known to be present on the surface of synaptic vesicles. And we asked, how did the vesicles behave? There was absolutely no change in the way the vesicles behave. There are 5 non-muscle myosins. We've looked at just this one, but we think this is relevant. We don't know the other myosins, whether they do something. And they just don't, you see a little bit less number of motion, number of vesicles moving is a little bit less. We don't know what that means, but the way they behave is exactly the same as they behave in wild type. So we have no means to assess if there is some change. If we saw something, then we could perhaps interpret it, but we don't see anything. And our interpretation is perhaps it's not the most important factor.
And the maps don't play any role in this?
So we have looked at one map, and what happens in these maps is the density of microtubules reduces. And therefore, we can't distinguish between reduction in the density of microtubules from what might be a map effect in vivo. Because for those, you need to do in vitro experiments, because there's so many factors that come into play when you look inside the organism. So that was not an easy experiment. Okay.